Hello everyone, welcome to 10-Minute Physiology. In today's video, we're going to be talking about how thyroid hormone circulates in the blood and how it's processed by peripheral tissues. So with that, let's give it a go. So when thyroid hormone enters into the circulation, it's actually going to bind to a number of different plasma proteins. Now, the first plasma protein that thyroid hormone can bind to is thyroid binding globulin. So thyroid binding globulin is going to have the highest affinity for thyroid hormone, and as a result of this, most of the thyroid hormone is going to be binding to this particular protein. The other two proteins that thyroid hormone can bind to are albumin and transthyretin. Now, all three of these proteins are going to be synthesized by the liver, and all of them can bind to thyroid hormone. Now, over 99% of circulating thyroid hormone will be bound to protein. However, it's the free or unbound hormones that are going to be responsible for biological activity. So therefore, this brings up a very important question. How do we calculate the concentration of free or unbound thyroid hormone in the blood? Well, the way that we can do this is by taking this equilibrium reaction here. So this particular equilibrium reaction shows us the reaction between thyroid hormone, thyroid binding globulin, forming a particular complex. So the T subscript X on the reactant side represents free thyroid hormone, T3 or T4. TBG represents thyroid binding globulin, and then TBG-T subscript X symbolizes thyroid hormone bound to thyroid binding globulin. So from this equilibrium reaction, we can give the equilibrium constant or calculate it. And what we get is this particular equation here. So next what I'm going to do is I'm going to solve for the concentration of free thyroid hormone. And when I do that, I get this particular equation here. So this equation is going to calculate the concentration of free T3 or T4 in the blood. Now, in order to calculate the concentration of free T3 or T4 in the blood, we also need to know the equilibrium constant for T3 and T4. So T3 and T4 have different equilibrium constants, and they're given right here. So by using this equation, we can calculate the amount of free T3 or T4 in the blood. So the next thing that I would like to look at is the purpose as to why thyroid hormone binds to proteins. So the first purpose is because by binding thyroid hormone to proteins, this is going to provide a large buffer pool of thyroid hormone in the blood. So what do I mean by this? So let's just say we have our equilibrium expression here. And let's just say that the body is using a lot of thyroid hormone, so the concentration of free unbound thyroid hormone in the blood decreases. When this happens, the equilibrium reaction will shift towards the left. And as a result of this, thyroid hormone, which is bound to the protein complex, will be freed from that complex. And by freeing thyroid hormone from thyroid binding globulin, this will help to maintain the concentration of free thyroid hormone in the blood. So this keeps the concentration of free thyroid hormone relatively constant at a minute-to-minute basis. So let's now talk about the other two reasons. So the second reason is that by binding thyroid hormone to a protein, the half-life of thyroid hormone is going to be increased. So in other words, thyroid hormone lasts longer in the circulation. And then the third reason is the majority of T3 is going to be produced by the peripheral tissues. And in order to synthesize T3, we need to have T4. So by keeping T4 bound to protein, this keeps a reserve of T4 in the blood, which is therefore able to be used by peripheral tissues to synthesize T3 from. And because T3 is the more active version of thyroid hormone, this is a very important thing to have. So these are the three main reasons as to why thyroid hormone binds to protein. So we're going to finish off today by talking about how thyroid hormone is processed in peripheral tissues. Now, different peripheral tissues are going to have different enzymes, and these enzymes are going to be responsible for processing thyroid hormone. So the first enzyme that we're going to be looking at today is going to be found in high concentrations in the liver, in the kidney, in the thyroid gland, and in skeletal muscle. So what does this enzyme do? So when T4 enters into the liver, kidney, thyroid gland, or skeletal muscle, T4 can be processed by a specific enzyme called a type 1 deiodinase. So type 1 deiodinases basically take an iodine off of the B ring and convert T4 into T3. So we see here that these tissues can produce T3 by using this enzyme. So this enzyme catalyzes the conversion of T4 into T3. Now this T3 can be further processed by type 1 deiodinases into T2, and then T2 can be further broken down into tyrosine. So what we see here is that in these tissues, type 1 deiodinases are going to be responsible for generating the most amount of T3 in the circulation. So the grand majority of T3 in the blood is going to be coming from these type 1 deiodinases found in the liver, the kidney, the thyroid gland, and the skeletal muscle. So there is another enzyme that can convert T4 into T3, and this is going to be the type 2 deiodinases. So type 2 deiodinases are very similar to type 1s. However, these are going to be found in high concentrations in the central nervous system, the placenta, and the pituitary. So the type 2 deiodinases can also generate T3, but most of this T3 is going to be for local use for the tissues. So there's one more enzyme that you should know, and this is going to be the type 3 deiodinases. So type 3 deiodinases can convert T4 into reverse T3, and it can also catalyze a few other reactions. However, we won't go over them in this video. So that's it for this video. I hope this video helped you understand how thyroid hormone circulates in the blood, and I hope it helped you understand how T4 is converted into T3 in the peripheral tissues. Thank you for watching this video, and I hope to see you next time.